Thank you everybody for joining us today at this webinar, combined webinar between IMI ConcePTION and LifeSaver. We have an agenda where we first start with two general presentations on the two projects, and then dive a little bit deeper on the specifics of each project. And I think we close the session with a panel discussion as well. So thanks for joining this webinar. We'll start with an introduction on ConcePTION now. And introduce myself first, of course. My name is Marjolein Willemen. I work in the medical policy team at Novartis in Basel. And in this role, I'm co-leading IMI ConcePTION together with Miriam Sturkenboom from Utrecht Medical Center in Utrecht. Can we go to the next slide, please? Yes. And just before we kick off, if you have any questions, please put them in the chat box. We are monitoring that and answer your questions at the end of the presentation, or connect you to the right experts that can connect with you afterwards to help you further. So IMI ConcePTION is a 5-year plus project. It's looking at the effect of medicines on pregnant and breastfeeding women and the outcomes for their babies. The whole project actually started with this very simple question on the slide here, which almost every pregnant woman and certainly every healthcare professional comes across. Can I safely use this medicine now I'm pregnant? Or is it safe to prescribe this medicine to my pregnant patient? And unfortunately, how easy the question also may sound, there's no easy answer to it. And there's not an easy source or a Google search that you can just do to help you to find this information, especially because I think it's critical for this type of questions that the information is evidence-based and comes from a reliable and trustworthy source in that regard. And that brings me to the next slide, where we have an example of a real case from a woman, we called her Amelia, who is suffering from severe nausea in her early pregnancy. She has been using an antiemetic that is known to be safe for use during pregnancy, but her condition is actually worsening, and therefore she and her physician are exploring other options, actually in order to prevent an hospitalization. Her physician is now considering to prescribe ondansetron, which is a more powerful antiemetic, but also recommends actually against the use in early pregnancy in the package insert. So what are now the options for Amelia? And where can she and her healthcare professional find this information that is robust, reliable, and evidence-based? Next slide, please. And these are exactly the questions that led to ConcePTION, because we believe that every woman has the right to safe and effective use of medicines during the pregnancy or during lactation, and it should be based on good evidence. And in nowadays world, with the emphasis on inclusivity and leaving no one behind, you really clearly see the relevance of this type of questions. And we know, of course, that during drug development, the emphasis is not on pregnant women. There's, of course, preclinical work done, but not all the animal studies are fully predictive of what is going to happen to women and their babies. And in general, clinical research, phase 1 to 3 studies, pregnant women are usually, I would say, almost always excluded, unless, of course, it's a treatment that is specific for pregnant women, such as investigating drugs for preeclampsia or something like that. And that means that most of the information that we get comes from the post-approval stage, when the drug is out there on the market and women do take these medications. And now it's our job to make sure that this information is indeed systematically captured, it's analyzed and used to generate robust evidence on the safety of those medicines during the pregnancy and lactation period. And if we can go to the next slide. There are a lot of numbers here, but no worries, I'll talk you through. So I think 5 million pregnant people in Europe every year. On a global scale, of course, the number is much higher even. And I think we can truly say that almost every woman, so 90%, uses medication during some point during their pregnancy or breastfeeding. And we also know that on average, a woman takes about 1.6 different medications throughout their pregnancy. And of course, I think it's clear that most women will wish to avoid medication use during their pregnancy, but we also need to acknowledge that it's simply not always possible. Some women have chronic diseases, such as multiple sclerosis, diabetes, epilepsy, where they could get seriously ill if they discontinue the treatment, or stopping could even have a negative effect on their unborn child. And of course, women have also acute illnesses such as infections or severe headache that needs to be treated. And as you can see on the right upper side of this slide, those numbers indicate that proper evidence about medicine safety is lacking. And if you look at the evidence, and we come back to that in a minute, it's also very inconsistent. That means that there is a huge burden of uncertainty, both among the women and their treating physicians and the healthcare professionals that are working with them. So you can see here that more than 70% of the medicines that are centrally authorized in the European Union, there is no information available on the use of that medicine in a pregnancy. And the number is even higher if you look at breastfeeding, going over 80%. And there's only 3.7% of the medicines that have an explicit statement in their labeling saying that it's safe to use it during a pregnancy or lactation. So that number is really very low. And now I move to the last part of this slide, which is the lower right corner. And these numbers are actually based on one of the first studies we did when ConcePTION started, was to look at the available evidence across Europe. And nowadays everybody goes online, so that's what we did as well. And I personally find these numbers really shocking, still find them shocking, because it shows that in more than half of the cases, so more than in 52% of the cases, the information about the safety of medicines during a pregnancy was inconsistent between different sources. And the number is not on the slide, but for lactation it was even higher. And that means that it is highly dependent on where you are looking and what source of information you are using. And that can lead to totally different information. One source saying, oh, it's fine, and please go ahead taking this drug when you're pregnant, to another source maybe saying, oh no, this drug is putting your unborn child at a very high risk, don't do it. And I think this inconsistency really indicates that there is a need for much better information for women and healthcare professionals, and also that this information needs to be consistent throughout and needs to be based on proper evidence. And that brings me to the ConcePTION project itself on the next slide. I can move one more. Thank you. Because what is IMI ConcePTION? It's IMI ConcePTION, so Innovative Medicines Initiative. We started in 2019 and we were supposed to end in March this year, but we have got an extension until the end of the year, the end of 2024. The project is co-led by Novartis and the UMC Utrecht and is funded by the European Union. We have the pharmaceutical industry also substantially contributing, not in the form of funds, but providing resources in terms of people and data. So at the moment, we have more than 200 people working on the project from more than 80 different organizations, which includes, for example, the European Medicines Agency, pharmaceutical companies, academia, public health organizations, and also people from the teratology networks in Europe as well. We are spread throughout Europe and also have some involvement of countries outside of Europe and outside of the EU as well. Next slide, please. And if you then look at ConcePTION itself, it's very clear that it is our goal and our mission to build a very trustworthy ecosystem, which Miriam is going to describe in a little bit more detail in a minute, which builds on the two pillars that I have just described. First, generating evidence-based information, monitoring the data that are out there, and ensuring that the information that we have is accurate, scientifically sound and robust. The second pillar is making that information available to the public, to the patients, to healthcare professionals, and everybody who is looking for information on medication use in a pregnancy and during lactation. Next slide, please. I think we can skip this one and go to the next, please. Yeah, and I think what one of the big learnings over the last 4 years has been is that if we combine forces and if we collaborate across the different parties that are involved, from academia, regulators, pharma, to teratology centers and healthcare professionals, we are able to make big steps, and we have made big steps within conception in developing such an ecosystem, and also in developing the best methodologies on how to generate evidence-based safety information on medicines that are used in pregnancy and lactation, and also how to bring this information to the public, to the pregnant women, and to the healthcare professionals in their daily clinical practices. And this is also where I would like to hand over to Miriam, and she will give you some more insights on how our conception ecosystem looks like.
Thank you very much, Marjolein. So maybe the next slide. So as Marjolein was stating, within the IMI conception project, our work has been focusing on two key areas. First of all, building that ecosystem. And we really think that we should not state that there is no information, because actually we should utilize the data that is being generated in healthcare and also complement that with primary data collection. But we need to look at the data that is available to learn and to provide best evidence. So we have been building a system to establish and validate methods that use this existing data and new data collection about the use of and the effects of medicines in pregnancy and also during lactation. Then there is this whole ecosystem that creates evidence, and then there is also this second key area, which is really about dissemination and summarizing all the evidence that is available in a valid manner, in a consistent manner. So this is done through the MUMS knowledge bank, which actually guides and advises those that need information, so the parents or healthcare professional, with consistent information that's been made together with a lot of parties that are involved. Next slide. So in order to, if we look at that first area, the key area, that's the generators of evidence, we actually distinguish three study networks. So first, the conception primary data collection. This is a network that collects information from women that are, or people that are exposed during pregnancy to a certain medicine or vaccine, and then follow them up over time during pregnancy and after pregnancy in the neonates, to actually learn also about immediate effects and about long-term effects, and we'll talk about that more. Then there is the conception electronic health record network. This utilizes actually data that is generated and captured, like real-world data, in the routine care system. So if people go to their general practitioner or to any healthcare professional, data are being recorded, and these data can actually be used in a pseudonymized manner to learn with all the proper safeguards and privacy regulations. And so this can be utilized as well. And then we have a third network, which is the lactation network. And this actually looks at the transfer of medicines into milk. And this is led by the University of Bern, Leuven, and Oslo. And we'll dive in each of those a bit deeper. Next slide. So the primary data collection, I think the key project there is the lifetime system, which enrolls women when they are pregnant and then follows through birth, but also then the neonate after birth at 6, 12, 24 months, 4 years, and 7 years. During pregnancy, the common data elements are being collected, and then after birth, the ages and stages questionnaire is utilized, and at 7 years, again, the lifetime questionnaire. So this is a systematic assessment of those pregnancies that are exposed to certain medicines by really actively enrolling the pregnant person. Next slide. Then there is the second stream on the electronic health record data. And so here you see a graphic on the bottom. This is like what we call a pipeline. Now the pipeline is modularized. As you can see it, we divide green areas and gray areas. The green areas in the pipeline are data sets. And then the gray areas are data transformation steps. And then on top and below, we have support of documentation and expertise. So that's more what is needed in the context to use this pipeline. So people and expertise, as well as tools to capture which data is being utilized, as well as protocols and statistical analysis plans. So if we go through this pipeline, on the left-hand side, we realize that for electronic health record data is available in many different places across Europe. And actually, it can be used for secondary purposes, but it's all in its original and local format. So that's what we call native data model. It's just the structure is all different, meaning different table names and different variable names. So the first thing that we need to do when we actually want to work together, which is what we want, is that we structure the data into a common structure. And that is what we call a common data model. And ConcePTION has developed this common data model for electronic health record data. So it's just a definition of how the tables and variables should be named. Now, then when you put everything in the same format, it doesn't mean it's the same, actually. So the content can still be different, meaning, for example, in the UK, diagnoses are being coded with a read code in the past, now maybe SNOMED. In the northern countries, they use ICD-10. In southern countries like Italy, they still use ICD-9. In the Netherlands, diagnoses are being coded in general practice with ICPC. So we have a lot of different vocabularies that are being utilized. And now all these diagnostic codes are in the same format, but it doesn't mean that we are talking about the same thing. So T2 is the step where we try to harmonize the languages, and we do that as part of the script. Then we create study variables, we apply a design in the T3, then we create analytical data sets where an analysis is being done. All of that is done by sending a script to the data, not the data to the script, by the script to the data in a distributed manner. And then only results are being pulled on a research environment and can be reported. So these tools have been developed as part of the ecosystem. People have been trained, and this is all operational and even adopted by other networks such as VAC4EU, the EU Pharmacoepidemiology and Pharmacovigilance Research Network, and the Sigma Consortium. Next slide. Then the third key area, the network, is about lactation. And here a lot of development has happened. When we started and wrote the project, actually very little was available in Europe in terms of expertise to do lactation studies. So we have built a lot during the project. And it started looking at in vitro work using memory epithelial cells to look at transfer of medicines in milk, potentially, then animal studies, porcine models, and then human lactation studies where women that are lactating were asked to donate milk in a biobank, and that was actually analyzed and then modeled for transfer. We'll talk about that more later. Next slide. So this is about the generators of information. Then we have those that are disseminating information, because in a proper learning health system, you should have the evidence generation, but also the dissemination. And in ConcePTION, this is called the MUMPS Knowledge Bank. It's Mothers Using Medicine Safely abbreviation. And this is a free-to-use open knowledge bank that can actually be searched on internet or your phone about drug use during pregnancy and lactation. Now, what is really the contribution of, I think, of the project is that many TIS sites, so the Teratology Information Services in Europe, were doing this in their local environment, but now we have developed a process where everybody is actually using a standard process to assess evidence that is out there and transforming that into a knowledge-based page. Our initial prototype of the MUMPS Knowledge Bank includes information on 40 drugs, and we, of course, intend to extend this to over 200 if there is future funding. Next slide. Also, what has been developed is an e-learning tool for healthcare professionals, and this is an online training resource about teratology and medication use in pregnancies that can actually be seen by healthcare professionals. It's also free to access and could become part of medical education. And if people would like to have access to this, please contact Brian Cleary. Next slide. So why we develop MUMS, and here you on the right-hand side, you see a screenshot. Here you see two different drugs and then an indication rheumatoid arthritis, and you can actually search those pages and look at the information that has been provided there. So we really try to become the primary pan-European source of information on the safety of medicines, and this is actually to address what Marjolein was saying, that currently there is information on internet, but it's contradicting and people don't know anymore what to rely on. So we want to become this recognized, reliable knowledge bank. Next slide. So as we heard before, the project started in 2019, it ends in December. A lot has been performed, many deliverables, many papers written, and a lot of tools tested. But we are building our future structure, where we actually want to champion the fact that we need a robust and reliable data and evidence on the safe use of medicines during pregnancy and lactation. So we have proven that this is possible, but we cannot only prove that we can generate information, we need also to advocate that actually this problem can be addressed, the knowledge gap can actually be addressed, but we do need a structured way of generating the evidence and policies that actually make this happen. So we are looking into what happened actually in establishing the pediatric investigation plan, the PIP in the past, where for children actually, which was also like a neglected population somehow, and now there is a lot of evidence being generated, and we could even think about a pregnancy investigation plan potentially. So we are advocating that this evidence gap is being filled. We want to galvanize our collective efforts of this very extensive collaboration and continue to further develop tools and platforms that are needed to both generate the evidence as well as disseminate this for the public good. Next slide. And I would like to thank you for listening to us and thank you for your interest.
So, hello everyone, and I'm happy today presenting the project LifeSaver, which is a smaller project compared to the ConcePTION, and we have less partners, but we share the common goals and ambitions. Is that what has been just presented? So in this short presentation, I will show you briefly about the project, mostly about the concept, the idea, the organization of the project, and then later another presentation from Thomas Zadrozny will be showing a little bit more details. How do we plan to go ahead with the exploitation of the results which has been foreseen in this project. So the starting point is quite similar to what was in the ConcePTION project. We see the evident gap of knowledge between the availability of information regarding the safety and efficacy, but mostly safety of the drugs and chemicals in pregnancy, and the lack of clinical trials for obvious reasons, and the lack of evidence, and also miscorrelation between different data, or let's say contradiction of the data which is available. They do not have a possibility to provide the sufficient information which women need to address or the clinicians or GPs needs to address, whether to decide whether the specific drug is sufficiently safe or risky in terms of pregnancy. So the concept of that in general, what we have, we try to introduce a new, I would not say the paradigm, but kind of concept, which we call the biodigital twin, comparing with a test method, which I will explain later in more details, but eventually it combines the proper in vitro and in silico platforms, which can be used for sustain of quality of life by providing risk assessment data. And this risk assessment data at the end of the project, which is planned now for the October next year, we already anticipate that we would like to have some kind of a pilot with proper interested stakeholders to provide a test and to see how this concept can be put in the clinical and pharmaceutical practice. So the challenge is what we have, and I think most of you know that about it already, that there are a lot of problems which is associated for using of the drugs in pregnancy. There is a lot of different ethical issues. There is a lot of different practical issues, a lot of risk associated with that one. And there is also different opinion whether the pregnant women can be used in the clinical trials or not, and if yes, to which extent, and maybe not. So it's a lot of discussion which is ongoing, but I think that most of all, let's say everyone agrees that there is indeed a lack of information for because we don't know to which extent especially at earlier stages of pregnancy, how exactly the drugs, how many drugs is actually going on or potentially could go through the placental tissues to the fetus. So we know all from economical point of view that the clinical trials are difficult, they're expensive, they take a lot of time, they pose a sufficient risk. And in the placenta, there is also an additional limitation because a human placenta is radically different from animal, not only in physiological properties or functionality, but also even in terms of physics, because the blood transport in the human placenta is different from the animals. So we can't just replicate the placental functionality in animal tests and that extent which could be potentially used for drug research or estimation of pharmacokinetics. So the focus on what LifeSaver is doing, we focus on the drug transport through placental tissue, which will be analyzed with a sufficient combined and integrated methods compared to the methods what we now used in clinical and in vitro practice, which produce quite variable outcomes, and they may have even less relevance to the real life. So what we can have as an example that for people who use who study pharmacokinetics, this is very familiar, that the typical assays which are used for investigation of the permeability of the drugs through different barriers. And as you can see, some of these barriers, they have cells, some barriers does not have cells, some are just synthetic membranes, they have only one time points, they have different equation. And one of the curiosity of this permeability assay is that different assays produce permeability which using the same units, but it's assessed by different equations. So when we have in the literature that permeability of certain drug is like this and this, but if it's not referred to specific assay, then its data is very much useless because we don't know how exactly this particular drugs test has been done. So, and the conventional analysis in placenta case is especially difficult because there is a lot of mechanisms. Some of them are not very well known, and there is no possibility to do it in vivo or in clinical trials to separate all this mechanism. That requires the use of very sophisticated equation from pharmacokinetics, which has a lot of unknowns, and there is a lot of differential equations to be solved without any good assumption how exactly parameters needs to be accepted and what kind of the solutions we need to use. So the LifeSaver goals basically are to create a reliable methodology to analyze and predict of transport of the substances through placenta, which is not possible to validate in humans. And as mentioned, that the biological twin system, biodigital twin system with a sufficiently high biofidelity is sufficient to be accepted for regulatory and healthcare and industry approval, which could answer the needs of drug chemicals to exclude potentially risky chemicals and drugs, or so it's kind of negative discrimination, which said that it is not safe or contraindicated in pregnancy, or positively, because there could be drugs which are exactly intended to be passed through placental barrier. And it's also important to know how they related to that drugs, what we know already. The concept of innovation of LifeSaver is based on the biodigital twin, which is combined in vitro and in silico system. And it's also linked within the project to the estimation of predictive possibilities for that, combining with the European policy advice and the regulatory science, which is all has to be combined together to lead to some application which could be realized in a clinical practice. So one of the technical things which is LifeSaver is considered is to eliminate the need for more complex and sophisticated equations, and to do it quite a simple way, because we can't go to toxicology before we know the exposure. So the idea of that is to know the exposure, to do the transport. And one of the concept what we have is a specific drug transport signature parameter or set of parameters which could be evaluated experimentally in vitro system, and also replicated in in silico system, which will replicate one in vitro system, and another one will be replicated as 3D dynamic structure of the placenta itself. So the idea is to make a simplified it with one model-free equation, which does not need to be assumed what kind of mechanism on it, or what is exactly, how exactly this happens, but just to compare the concentrations and the concentration dynamics on two sides, on maternal and fetal side of the placental tissues. The approach of the project is to combine an iterative process, starting from the pregnancy biology as a boundary conditions, to go through the tests, in vitro tests, in silico test, and combine a regulatory signing, EU policy advice, and the clinical relevance to or result in some integrated solution which could be potentially used for drug development or drug repurposing, or also to estimate of the risk of the drugs which has been known already on the market, but from that information of the safety in pregnancy is missing. So regarding the risk and quality and assessment actions required for the drug development processes, I think that there is a risk. What we try to address it, which kind of them products of drugs and chemicals could potentially pass the placental tissues, and they could create serious effects, adverse effects on unborn children as well. And also there is no reliable methods to get this risk assessment data, because there is no animal models which can replicate the placental tissue in a controllable and cost-effective way. So in terms of the risk assessment in LifeSaver, we don't consider toxicology as such, because our project is also focused on the drug transport mainly. So when we know the exposure, then it's another step in toxicology and teratology to see how much this potential concentration or exposure in time could create potential effects. So the vision is that, of course, very similar to conception. So we totally agree that every pregnant woman has to have a proper live in safe environment, and has to be protected not only from the drugs and pharmaceutical products, but it's expand within the Green Deal, European Green Deal concept also to all chemical products. We talk about environmental chemicals, we talk about household products, cosmetics, and everything which is potential can be interacting with the placenta. So our vision is that when the system will be operational, it could be de facto and de jure a standard for preclinical risk assessment for pregnant women for potential exposure, and it can be eventually deployed worldwide. So we not talk only about European market or European health space. The consortium of the project is we have 17 partners, and you see it on the map there, how they are located around Europe. We have quite a lot, maybe more than half partners are SMEs, small and medium-sized enterprises. We have universities and research organizations, and they represent quite a lot of spectrum of the knowledge and competence from pregnancy biology to cell research and to up to policy making and dissemination. So it's a very multidisciplinary consortium which try to solve this kind of problem. So with that one, I thank you for your attention. As you can see here, the website, which you can see more information and some video materials. And just to remind that at the end of the project, which is November 25, we would be very happy to cooperate with the people who are interested to test the system in order to start with some kind of pilots for specific drug transport. Thank you.
Okay, so first of all, thank you also for the opportunity to contribute to this very interesting webinar series today. So I will build a little bit on the introduction also that Miriam already has provided, and also give you some overview of what we have been doing within IMI Conception, especially the work packages 3 and 4. And I do want to emphasize that this is the result of the work of many people, many parties. I'm just speaking on their behalf here today. So what I will be discussing is the non-clinical and clinical approaches that we have developed and used to determine the secretion of medicines to breast milk. So in terms of IMI conception structure, I don't think I need to spend a lot of time here, but just mention you that we are here referring to work package 3, which were the non-clinical approaches for lactation, and then work package 4, where the actual clinical lactation studies and the biobanking was done. To start maybe with the clinical aspect, so in order to study the extent of passage of drugs into human breast milk, you can of course do clinical lactation studies. This is quite challenging because of course the population where you have to do these studies is a very special, a very vulnerable population. However, I think the good news is that within IMI Conception, several of these clinical lactation studies have been done. Here you see the example of levocetirizine, also cetirizine. It's essentially the same drug where actually the transfer of this compound, this drug, has been studied during lactation in breastfeeding women. And so you see here a typical concentration time profile. So what you would typically also see in plasma, but in this case, you see it in milk, that you have a peak concentration that's being reached, and then gradually over 24 hours, the drug is disappearing again after this single dose of this drug from the milk. Of course, if this drug is appearing in the milk, that means that also the infant that is breastfeeding can get exposed. And that's, of course, the type of information that you want to generate here. What is very important in this context is that the protocols that you can use to generate this information, they have to be very well thought out, and you have to consider a lot of ethical aspects. This is just another example of a protocol that was recently published. It was in collaboration also with the group at the University Hospitals in Leuven. We called it Umbrella Study Protocol, which is kind of an umbrella protocol that was developed, which can be followed to study the transfer of different drugs in breast milk. This was a very, in a way, I could maybe say opportunistic approach. So we were able to get this approved by the ethical committee. That means whenever women were available and were willing to actually donate a few milk samples, that we could take the opportunity to measure the drugs in the milk and to have some information that in many cases for many drugs is otherwise not available. So please also go to this very recent publication if you want to read more about that. Before I dive into the non-clinical approaches, I just want to mention for the clinical lactation studies, what you can consider a primary outcome is the so-called milk plasma ratio. And it's actually the exposure that you have for the drug in the human milk divided by the exposure in the plasma, so the systemic exposure. So it gives you a relative idea how much exposure there is in human milk compared to the exposure in plasma. Exposure in plasma, which is commonly known in most, of course, for all drugs that are on the market. So, but if you have this milk to plasma ratio, you can make a lot of predictions about the exposure and can start to make assessment about potential safety issues. So what are then the non-clinical methods? And that's a bit the focus of work package 3. And the focus of this presentation will be a little bit more on that part, because it was also the part that I was personally more involved in. It doesn't mean it's more important, but I can just tell you a little bit more about that. And of course, we can build on that further during the discussion and the questions. So what kind of non-clinical methods can be used, and how can they help to fill what you can call the data generation gap regarding medication safety during lactation? And then here in this case, we're talking, if we say non-clinical, we're talking about in vitro models. I'm referring back to what was just mentioned also in the previous presentation, where actually you can mimic also the blood milk barrier with these mammary epithelial cells. You can use bottom-up in silico models. We call it physiologically based pharmacokinetic modeling, and that actually refers back to what Michael was also discussing, the biodigital twin concept. And then you can also use animals in an in vivo, complete in vivo setting. This is kind of the graphical abstract for these non-clinical methods. Of course, on the far right, this is the kind of information that we want to generate. As already indicated in the beginning of this presentation, you can use a clinical lactation study to generate that information. However, of course, that is quite challenging, and it will take still a lot of time to generate that information. And we should, of course, continue to do that. It will take a lot of time to generate that information for all drugs on the market. Essentially, if you dose mothers during breastfeeding with the drugs that they need, some of the drug may end up into breast milk. That's what you can measure in the milk samples. But you could also consider to extend your study design to also do opportunistic samples, for instance, in the infants after breastfeeding, and to see to what extent they have some of the maternal medication in their blood. So in order to combat to that information, indeed, you can take the clinical approach, but you can also take what we call two non-clinical pathways. The first pathway is what we call the in vitro in silico pathway. The in vitro model is what also was presented in the previous presentation, or at least in terms of concept, where you can measure concentration differences, or the extent to which compounds are able to pass from one to the other compartment across a barrier of mammary epithelial cells. So the blood milk barrier, you basically mimic that in vitro. And that kind of information can then be incorporated after scaling, after extrapolation, in this physiologically based pharmacokinetic model, where you actually combine physiological information about the population. As for instance, you also described, apart from the different organs, you also describe the breast milk compartment, and that compartment then directly feeds in, so to speak, into the infant PPK model. So in this way, you have a complete digital representation about the maternal and the infant physiology, and you can conduct, let's say, virtual clinical trials in these populations. And that would allow you also to generate, to predict this milk plasma ratio. Of course, you do need some measurements for drugs, at least in this point in time, via clinical lactation studies, in order to be able to validate this approach. Another part that we also have explored as part of work package 3 in IMI conception are the in vivo animal models, where the Gasik and mini pig was selected as animal model. And I will just show you some data later on. Actually, this was actually quite successful in predicting the milk plasma ratios. The milk plasma ratio was determined in this animal and was quite predictive of the milk plasma ratio that had been determined for the same drugs in humans. Of course, we're still in the process, and you see here also cross-validation to leverage as much as possible the advantages, but also the limitations are there, but the advantages of the different model systems to strengthen the predictive ability of these non-clinical pathways. So a little bit of text here to just give you maybe a little bit more background about what kind of molecules did we study. The focus was on small molecules, and the focus was really to quantitatively assess the passage of medicines across this blood-milk barrier. And in this way, then it was possible to assess the exposure to maternal medication. Important, of course, whenever you use non-clinical methods, that you can translate that to the human situation. What is the exposure in human milk? And then further on, what is the exposure in the infants? And that could be done with the in vivo animal model, but certainly also with the biodigital twin, with the PPK model approach. And the anticipated outcome of work package 3, and that's where we are more or less now, is really a proof of concept that we have generated for what we call a non-clinical platform, because you have different model systems for exposure-based medication safety during lactation. Initial verification, as is mentioned here, has already been done, because luckily we do have some measurements from clinical lactation studies which can be really used to validate the approaches that we have developed. We have been in contact also with regulatory agencies trying to get qualification advice. It's a little bit of a long story where we are now, but I can definitely give you some feedback maybe based on questions later on. So in terms of compound selection, we have focused on small molecules, also molecules, drugs for model drugs for which clinical reference data were available, because otherwise you cannot validate what you're doing. We did look at diversity in physicochemical properties in terms of lipophilicity, to what extent these compounds were ionizing at physiological pH. In terms of pharmacokinetic or ADME properties, are these drugs mainly cleared by the liver or the kidneys? And this kind of diversity was built in the compound selection. And you can see here on the right, these are just the lists of the compounds. So most of the compounds that we have tested. For the in vitro and the PPK model, you have more compounds because they are higher throughput than the in vivo animal model, but still all the models have their advantages and limitations. So it was important to have them side by side. I'm not going to read maybe all the technical details about the animal model, but let's say the conclusion here for selecting this model system was that as compared to rodent models that were currently used or until now used for lactation, the gutting and mini pig was really the most optimal choice in terms of in vivo animal model because of the physiology of the milk glands, which is much more comparable as compared to other species to the human physiology in that respect. The study design was to actually sample both the sows and the piglets for a period of about 3 weeks after also a week of training. So this was quite an intense type of study, but also generated high resolution data for drug milk excretion. So also the kinetics really for these different compounds that were tested could be extracted like this. This is just an overview of the drugs that were tested in this in vivo animal model, including amoxicillin, venlafaxine, metformin, and levocitrazine. And you will recognize some of the compounds were also already tested in clinical lactation studies, which of course makes it interesting because you can compare afterwards. What you will see is that in most cases, and venlafaxine is an exception, the plasma concentrations are higher than the milk concentrations. Levocitrazine, it's maybe a little bit more break even, but for venlafaxine, the milk concentrations were higher than the plasma concentrations. So this was more of a lipophilic compound that is accumulating into milk. We also had the opportunity in these in vivo animal models to measure in the piglets, and what you see in actually for all the compounds, and that's actually good news, of course, for drug use, medication safety during lactation, that even for venlafaxine, the concentrations that we found in the plasma of piglets, and that was also the case for metformin and for amoxicillin, that these concentrations were much lower than the concentrations that we found in the milk. So indicating that the exposure in the at least for the compounds that we tested in the piglets in the system was actually very low, and that can give you some guidance in terms of safety of these compounds. I think one of the most interesting results that we got here is we calculate the milk to plasma ratio in the animal study for those 4 compounds that have been tested in vivo in the gutting and mini pig, and compared that to the reported milk to plasma ratios in human from clinical lactation studies, that we found a very nice correlation. I think we can probably say that we have been a bit lucky. It's only 4 compounds, but still I think it's very promising about the capabilities and the predictive performance of of this animal model. There's some more information about the metabolite of venlafaxine, which is the O-desmethylvenlafaxine. It's an active metabolite. Just to mention too that the in vivo model also has the advantage that also metabolites can be covered in the evaluation. Okay, that brings me then maybe further to the in vitro model to tell you a little bit more about that one. I think maybe I'll need to speed up a little bit in time. I don't know, Marjolein, how I'm doing time-wise.
I think you're good.
You're okay. Yeah.
Okay, yeah. Thank you. So for the in vitro models, essentially the hypothesis where we started from is that that we considered that the blood-milk barrier, and actually especially these mammary epithelial cells, are the main barrier, the determining barrier for the determining factor, if you want, for compounds to cross into the milk or rather stay into the plasma. So that's why we decided to try to make cultures of these mammary epithelial cells, and it was after a while quite successful. And we were actually able to do this, and it's not unimportant in the context of this project to do this for human mammary epithelial cells, but also for the mini-pig mammary epithelial cells. And the original objective there was, well, if we can do this also for the mini-pig and we have high resolution in vivo data for the mini-pig and we have the in vitro mini-pig model also, that would allow us to much better cross-validate and to let's say, validate the what we call the IVIVE, the in vitro in vivo extrapolation algorithms. We also are still in the process, and we are actually collecting the data as we speak to enhance the biorelevance of these in vitro models by, for instance, using diluted milk as the medium on one side, of course, of the cultured cells. These are just some background characterization data. This graph just indicated if you keep these cells in culture, you have to have a little bit of patience for about 1 month, that actually the electrical resistance across this barrier is increasing, this transepithelial electrical resistance, which is an indication for the fact that these cells also in vitro manage to form a biological barrier that has resistance to transport of ions, and also will then represent the biological barriers for compounds that you put on them when you want to measure the transport. Also the same for the mini-pig mammary epithelial cells. There, the nice thing is that for the mini-pig cells, you were able to achieve this functional model in about a week, where it took up to 4 weeks for the human model. So that's, of course, also a species difference that we had to deal with. I guess the most important aspect here in terms of the project that I'm discussing today is that if we looked at the permeability coefficients of about 10 model compounds with diverse physicochemistry in the human mammary epithelial cell model, we did see quite a broad range in permeability coefficients. So we have more hydrophilic compounds showing much less transport, whereas smaller and more lipophilic compounds, you see that venlafaxine also shows up here again at the higher end. The one that was accumulating in the milk also has a higher permeability coefficient in this in vitro model. Of course, you have to also consider the limitations here. I think it was also the message that Michael was bringing before for the placenta. This is just a very simple in vitro model. Of course, these cells are there, but that's the only resemblance that you have with in vivo. So you have to translate this kind of information and combine that with in silico approaches to translate that into a decision or a prediction that you can make for the human situation. Also for the, and that's not surprising, I guess, for the mini-pig mammary epithelial cells, you were glad to see that we did see a comparable kind of range of permeability values, and so that we were able to distinguish between compounds that showed higher versus lower transport across these biological barrier. We're still in the process of processing these data, implementing them in the PPK model, but we are quite hopeful that feeding this biodigital twin concept, the PPK model, if you want, with these in vitro data, will have an advantage in terms of predictive performance towards the human situation. So I talked about the in vivo animal model. I talked about a little bit about the in vitro cell culture model. I do need to give you a little bit more information about what we did in terms of PBK modeling. So what is PBK modeling? And this is again the same concept as what was referred to previously as the biodigital twin. Essentially, you combine physiology, pathophysiology of a human being, of a specific population. It can be an infant, it can be a mother during lactation, with pharmacokinetic properties, call it ADME mechanisms. To what extent is the compound absorbed, distributed into certain tissues and also in the milk. And that allows you to predict in a bottom-up way, based on de novo principles, first principles, what is the concentration time profile of these compounds into the blood and even also into tissues or into other body fluids, including the milk. This is one of the oldest schemes that I know for PPK modeling, but still a very valid one. Essentially, a PPK model consists of two components. You have a system component, which is the physiology or the pathophysiology. This is all the organs that you have, the weight of these organs, to what extent is blood going to these organs, for instance, and then also the drug-specific components. These are the pharmacokinetic properties and also the physical chemistry of the compounds. Together, that forms a PPK model. And the nice thing is that you can then study, having such a PPK model, you can study intrinsic and extrinsic factors, and what is the influence of those factors on the actual pharmacokinetics. An example of an intrinsic factor is there a state of pregnancy or lactation? That means that actually your physiological component will change, and you can study what is the impact on the outcome, the pharmacokinetics that you will predict. You will see that there's numerous applications already nowadays for PPK modeling. Predicting the pharmacokinetics in the milk during lactation and also the pharmacokinetics then in the infants during lactation is one of the major applications that we have been dealing with. Before I go into those details, let me just also mention that one of the advantages of PPK modeling is that you can tune the resolution to which you include certain mechanisms. One of the typical examples is that at each organ level, you can decide whether you adopt a permeability limited model versus a perfusion limited model. To what extent is it actually the blood flow, or is it actually the penetration of the compound in the tissue that is going to be rate limiting in your overall disposition process? And of course, it also means that you have to have that information, but it can make this approach very powerful. This is the general workflow that we followed for a PPK model specifically for the lactating population. You always start from a, let's say, a standard, a reference PPK model, also where you have a lot of clinical data available for verification. I mean, it's still a bottom-up approach, but you will build these models for healthy volunteers, and then you will extrapolate it by changing the physiology, for instance, to the maternal, the lactation PPK model, or to the pediatric PPK model. And that can be children of different ages, and depending on the population you are interested in. To make a very long story short, what you can do, you can actually calculate what is the concentration that you find in the blood of the child, and you can also calculate what is the dose that you have delivered during maternal taking of maternal medication, what is the dose that is delivered via the milk to the child. Of course, making certain reasonable assumptions, what is the volume of milk that is taken on a daily basis per kilogram body weight, to give just an example here. In terms of PPK model performance, we went through these different steps through this workflow. This is just an overview when we compared the predicted peak concentrations with the observed peak concentrations for about 10 model compounds, and you see each dot is actually another study and another drug. That in terms of predictive performance for the reference models, we were doing very well. Also for the exposure, you see here AUC instead of peak concentration, that's the right panel. This was also satisfying. And then we actually extended these models to the lactating population. In each case, you can see the point, which is the reference value, what was measured in a clinical lactation study, or actually the point is what was predicted. Sorry about that. The point is what was predicted, and then the range is what has been reported in clinical lactation studies. You will see that, yeah, because of the limited information available from the clinic, sometimes that's a very broad range in the milk to plasma ratio, but in most cases, we were actually hitting the range that was also reported based on clinical studies. Of course, these models need to be further optimized, and we also need to improve the genericity of these models, and because now we have built compound specific models, but at some point you want to gain sufficient confidence in this type of PPK modeling framework that you can apply it for every new compound, even when there is no clinical lactation data available. These data also allow you to, as I quickly mentioned before, to calculate the relative infant dose, which is a relative indication of the percent of the dose that is reaching the infant relative to the maternal dose. And you see that for most drugs, and in a way that's actually known, but not for all drugs, but for most drugs, these relative infant doses are very small. Of course, then you still have to assess to what extent is this potentially still an issue, a safety issue in the infant. But the nice thing is that you get quantitative information that can help support your decision-making regarding safety during safety of medication during lactation. That brings me to the conclusions. So the clinical lactation studies, what I mentioned in the beginning, of course, yield unique quantitative data regarding concentrations of medicines in human milk. Even with a very limited number of measurements, this can be a game changer, especially when combined with PPK-based simulations, and what I presented in the latter part. I think we also can conclude that the developed PPK models resulted in reasonable prediction of human milk concentration time profiles for at least 80% of the tested medicines, and we are improving this further as we go along. Then we also have the in vivo animal model that I presented, which is based on the gutting and minipig, and it seems that it may represent the gold standard species in case of in vivo lactation studies need to be done before in a preclinical stage. And so the milk to plasma ratios for the 4 compounds that we have studied correlated very well with the milk plasma ratios that were reported previously for the human. The in vitro model, I think there's still also some work to be done there, but the nice thing is that we were able to distinguish between low and high permeability coefficients for these different model compounds. Of course, these in vitro and vivo algorithms need to be developed further, and we need to see how we can use these permeability data, permeability coefficients as input in the PPK models. At least we think that within IMI conception, we have pulled this this domain forward and actually started to make a proposal to generate a proof of concept so that it's possible to generate quantitative pharmacokinetic data specifically in these populations, and that in a very standardized manner. Before I end, I just want to very quickly then cover what is next. So we have already a name for a follow-up project, a follow-up consortium, if you want. It's called Milk for Baby. You see the different names of the partners that are already there. This is very preliminary. I want to add that, but where we want to focus really on recruitment and sampling. So to what extent is it possible to recruit patients and to be able to access samples, and because when we have these samples we can generate this information. So this is about the clinical lactation studies. How do we store them? We need protocols for that, and that needs to be further standardized, and there also a network covering these efforts needs to be established. We need to further focus on the bioanalytical aspects according to the highest quality standards, also the pharmacometrics aspect, modeling, whether it's top-down or bottom-up modeling. All these technologies that are advancing very rapidly needs to be leveraged. And of course, it's very important to then disseminate that information, go into interaction also with regulatory agencies, something that we have also done for the non-clinical methods recently, and see what we can also learn from there. And that's also very important. I have some further text about Milk for Baby, but maybe with respect to time, it's better to give it a little bit of time now for questions. And I thank you for your sincere attention.
That after these 3 presentations we have seen already, it is very clear that ConcePTION and LifeSaver are sharing quite a lot of commonalities. We have identified the same problems, we have identified the same barriers and gaps. We are almost going into the same direction concerning on how technology and science can help us to solve those problems. So I think it's an important point that with which we will be able to further detail the collaborations will be coming in the future. Concerning this specific presentation, as Michael said before, We are rather an integration project. We have integrated different technologies and different scientific knowledge into a solution for a specific problem. So it has been an integration, a technology integration project. And of course, we are seeking that this will have an answer, a solution for the problems we have identified, which are exactly the same ones we have been talking since the beginning of this workshop. So in the exploitation activities of LifeSaver, we have simply asked ourselves these specific questions. What's the problem? What are the barriers? What technology could solve the problem? We have, of course, to do different experiments and validation activities to see if that's really working or not. And finally, we have the other barriers that are to convince stakeholders, regulators, industry, or whatsoever, that our solution is good. And that's basically what I'm trying to present you right now in this presentation. So what's the problem? Well, the problem is the same we are talking about since the beginning. I think I don't have to be more specific. There are percentages of problems outside there which are fully related to mothers in pregnancy and babies, specifically concerning their health. So one of the driving factors we have found in our project is that there is a higher maternal age. Of course, we have the environmental pollution situation nowadays, widespread medicines used during pregnancy, and the lack of suitable pregnancy complications treatments, which there are more and more. It's increasing. That is the principal characteristic of the problem we are analyzing. All this data, all the complications, they are increasing from one year to the other. So what are the barriers? What are the gaps? What exactly we find which is making possible that the problem remains there or gets worse or we don't find the solution? It's clearly a problem of gaps. For example, we are missing reliable methods to identify and address the exposure of the risk in the first pregnancy trimester in the case of LifeSaver. And the risk assessment data are missing. The animal models are not completely to replicate in many cases, specifically concerning the placenta. And of course, we also can see that there is a lack of artificial systems that could mimic the placenta tissues. As we cannot go to the placenta in any moment, we would need to be able to understand it not in vivo. These are the barriers we have seen from the problem we just were exposing and discussing before. Well, in LifeSaver, of course, we have a vision, we have a solution, and we try to explain what is the added value. Again, I would like to say you will see there are a lot of similarities between ConcePTION and our project. So in the case of LifeSaver, as we are very placenta specific, you should not see our presentation as narrow. Contrary, it could perfectly adapt to the more large scope which has your project. So finally, the vision, of course, as Michael explained before, and most of the presentations are explaining constantly, is to have healthy mothers, healthy babies, and that can last long time. Why not for generations and generations? Pregnancy is a very special moment in the life of humanity, in the life of a woman, and in the life of the babies coming up, as it's the moment where transmission is taking place and is also ensuring what will be the future health of the babies are coming into our world. So we need to understand better what's happening there, and we must ensure, once knowing what's happening there, that the health quality of mothers and babies will increase which, or at least in the first stage, to stop the issue that the problems are growing from one year to the other, as I said in the first slide. So how does it work? That has been explained already before by Michael, much more in detail, but basically it is a new alternative method platform that provides objective risk assessment data that has been, the lack of data has been one of the basic points of everything has been exposed today, for example, and that we know. And of course, in our solution, we would not need the clinical test on pregnant women, and we don't use animals for that. So it's a concept where we put together in silico and in vitro in a way of a biodigital twin in order to produce this risk data. It was much more explained in detail before. So finally, we're having a problem, knowing the gaps, and having a solution. What's really the added value? What's happening outside there? In LifeSaver, we have been analyzing for the last 3 years, what does it really exist outside there as a solution, as a technology, or as a protocol, or whatsoever tool that could help to solve this problem. And we get to the conclusion, and I'm sure ConcePTION as well, that there is not much done outside there, and there is a lot to be done. So it is very clear that to convince the other stakeholders of what's our added value, it will be slightly easier, we hope, but the problem will be to make them understand perfectly what is exactly the problem and the solution. So we are talking a LifeSaver of a scientifically based tool for experimental screening of chemicals and pharmaceuticals, specifically for the risk crossing the placental barrier. Amazingly, when we were talking of lactation just before, there we had exactly the same replication issue, but concerning the blood and milk barrier. And in the same way, we know that in the human body, we do have many other barriers. So we are talking of a specific science and medical situation where it is important to understand how this crossing is taking place, how the transport is being done, and what are the consequences on both sides. So what we try is to understand what are the potential effects on pregnant women and their fetal health. We are seeking to have a regulation improvement, and we also try to have a better risk assessment system, and of course to have access to much better data, which can be useful for different stakeholders. That is specifically the added value we are proposing. Clearly, when we are talking of risks and drugs or chemical products going through this placental barrier, we are also completely in the environment of regulation, in the environment of policy, and therefore we are trying to get our LifeSaver solution into the European policy first, and get it also within the European regulation as a consequences later on, after being understood by policy and all the stakeholders. What we really, what's our real goal? We seek to have that any, you can read it on the right, that anything that's produced or imported, sold in the European Union, or including exported outside by European industry, is evaluated, and that the risk of this is known, so that we can ensure a better pregnancy and a better quality of life of babies, children in the future. Risk, we try to minimize the health and environmental risk in a proper way. The stakeholders that we are trying to put together, of course, are the policy and regulation stakeholders, but it's also the industry, it's also the academia world, those who are working in research concerning the subjects we are talking here about. And we try to put all these stakeholders together in order to get through our message. So today, what are our next steps? With the industry, we are trying to customize our model within their operations and strategic vision. We are sure we have an added value for them. I'm also sure that ConcePTION has an added value for them. The other research stakeholders, we are trying to understand with them what is the applicability of our model in other organs. We could see that just in the presentation just before this one, there are other organs, other pathologies, therapies, and medications to which this could be applied, but it needs more research. And finally, policymakers and regulators, the idea is to upgrade the scientific base and information, as well as increasing the quality of regulation at a very early stage, and that can take into consideration faster any emerging issue. Today, we try to have a closer contact with these three groups of stakeholders, and also we have the aim to team up with other partners for a future co-proposal in order to ensure future funding in a way that the different elements that still remain open can be developed and find additional or deeper solutions. Okay, that's basically the exploitation strategy and I would say the idea behind LifeSaver once we try to get in touch with the people who have a need to use this.
Thanks for the info. We're a little bit ahead of schedule, so I think that's good. It doesn't happen a lot, but gives us some extra time to discuss. Can I ask all the speakers to turn off the camera so that we can have a joint turn on the cameras to have a joint discussion. And in the meantime, allow me to shortly introduce myself. My name is Jasper Levink. I'm a biomedical engineer by training, and have been working on the interface of science and business for most of my career. And I was involved in the very early stages of the project. Actually, Miriam and I sat together to craft the project plan, I don't know, 5-6 years ago already. And we're now working towards the end of the project, really looking at the sustainability and also, okay, what is the future impact of the project and how can we really realize that? And that is also what we would like to discuss a little bit with the people that have been presenting today in a short panel discussion. And I think Thomas already nicely described it, and Miriam confirmed that there's a lot of overlap in the identified needs, and also complementarity in the methodologies that that both projects have developed and presented. And I think Thomas already described shortly how he sees the future, but I'm interested to hear all of you in the sense that so how do you see the synergy between the projects, and how do you see that these complementary platforms also synergize in the future? Anyone who would like to start, or shall I just give some people the floor one by one? Michael, would you like to start?
If you can a little bit make a little bit more focused what kind of questions you have set up.
I wanted to start a little bit broader, but let's start with Miriam or Marjolein on how you see the synergy between the projects in the future. Miriam?
Well, I think that is a thing, because you know when you start with 2 wide questions, you will get wide answer, and it will take some hours.
Absolutely.
But in order to keep the time, I think that this is one of the reasons why we have approached the ConcePTION project, because we have seen the synergy in terms of the goals, in terms of the data gaps, in terms of the added value. And I think that that what you mentioned about this overlap, I see it more like a cooperation possibilities, because with the scope and the spectrum of partners of ConcePTION, and that specific part which LifeSaver is aiming to suggest to the stakeholders, because we, as you have seen from the presentation, we focus only on some small part. We like to demonstrate that how we can measure the transport of the drugs through the first trimester placenta, because that is, you cannot do any other way, and you hardly even can validate it clinically ever. So this is a challenging point, which is good. But what we can see is that I think that what we can do together is basically to complement what Thomas has mentioned as well, in terms of the development of the proper hybrid methods, whether you call it new alternative methods or new advanced methods or NAMs for general, and to say that how we can utilize a modern technology to get best of the experimental data what we have, and to create a data which could be potentially accepted, or not only by industry but also by regulators, and which eventually will result in that what Marjolein was shown in the beginning, in the instruction for use, where the doctor and the patient can basically read it and said, okay, this is a green risk or yellow risk or red risk, whatever, should I take it or should I not take it? Because at the end of the day, whatever complex technology we have, in respect to the user, it has to be sufficiently robust and simple. That is a kind of more engineering approach to that one. And I think that the utilization for what we have done in both projects, we're still in a mid-term in LifeSaver. I think that we have a lot of potential.
Thanks, Michael. Miriam, you want to
Yes, so
add your perspective?
yes, so I think we can do things on multiple levels now. So first of all, I think if you look at it from a methodological scientific perspective, which is, tools that we utilize, I think they are very complementary and almost feel like a pregnancy trajectory, from early exposure or early prediction to lactation, to follow-up, and etc., long-term follow-up. So I think from on a methodological perspective, we do see like a combination of tools and methodologies that are NAM, you call them, new advanced methods, appearing. And you could line them up very nicely, I think. I don't know whether then it's complete, like whether we miss pieces, whether reprotox would still be needed, for example, if we look at the old methodologies, but we can talk about that. So, but on a tool, I could see that happening. I think what I'm hearing, and I hear your need in terms of like, we could also liaise on the level of stakeholder engagement, no? Because collectively we have a lot of stakeholders on board and potentially able to make some influence towards a certain direction. So I think on those levels, and probably there are more, but it would be really good to connect, because each of us, we are struggling to be heard, no, in a way that this is actually changing the landscape, and I think what we are showing is that we have access to the data, we have access to the methods, we have access to people, and there is readiness to really change it. So I would like to think of how we can formulate a solution that could be embraced by the policymakers, no? Like a proposal for a solution rather than describing the needs, but make a positive change proposal. I think that's what I'm seeing. So I'm very enthusiastic, actually, Michael and Thomas. Yeah.
I think that what a short comment based on that, it is exactly what was a driving force for LifeSaver at that time when we write the proposal a few years ago. And I think the ambition, what we all have, and we share it with a ConcePTION for sure, is to saying that the solution we're going to suggest to the policymakers, it's not obligatory, has to be a new directive or a new regulation, a new REACH regulation or whatever, new pharma, but it's basically an improvement and a small step ahead for that one to, for example, that all the pharmaceutical or drugs or chemicals, whatever they use, they need to be assessed for the risks. So this policy is already in place and it's already being enforced. And I think that is what we can suggest also the technical solution, methodology solution, which could be potentially translatable to any on the players, not maybe what they call it stakeholders or whatever, but basically that which could be practical. I think that is one of the on the solution what we can offer.
Adding to that, Michael, I would like to mention that clearly the resources, the know-how and the expertise is there. If you put together both consortia, it is very large and the expertise is very high. So I think next steps are rather integrating that group, interest group, or call it whatever you wish, and defining 1, 2, maybe 3 directions and actions for each of those directions. Clearly, one of them is the policymakers. Remember, what they want is representativity, and we can provide that. And what they need is to have better information. We can provide it, and they want to know how far technology can solve the problems we are trying to solve. And they will have that. So at the end, we are talking of a communication problem. They don't know it yet, or they don't know it correctly, or they don't know much about it. And that's where we have to put our power together in order to communicate that as of September on, when we will have new policy made, at least at the parliament over there, and try to convince them that this specific situation incredibly has not yet any representation nowhere. How it's possible that feeding and having babies is completely a bit at the side of any European orientation. It's important. And for that, we need to have people who want to take that representativity. And I think both of our projects and all our stakeholders around are the perfect mix to do that.
Yeah. So I think, so what Miriam, you're suggesting, right, a solution, and what you're also saying, Thomas, we need to come with 1, 2, or 3 key actions or proposals. Marjolein, I can ask you from the industrial perspective, what kind of what those solutions could look like if you take it from the pharmaceutical perspective.
Well, I think that's a very interesting question, and I think from my perspective, being in the pharma industry at the moment, but also having worked in health authorities for more than 10 years. So I've really seen both sides of the coin, so to say. I really do think that if you want to advance the information or the evidence generation on this topic specifically, that there is not one side that can do it on its own. So it really needs to be kind of a collaborative effort from both pharma and also regulators to see what methodologies we have generated in those 2 projects. How can we bring that to a next level and integrate that into new policies, new guidelines? What can be asked from companies to be done? What can companies provide? What is possible? Not only focusing, I think, on new medicines, but also, and maybe equally important at least, is drugs that are already on the market, where maybe there are generics available, where there's more exposure, where things can be done. as well. So I think that is really going hand in hand. And I do think that if you look at, for example, what has been done with the pediatrics, there was an incentive for companies to start doing it. I think for if you look at pregnancy-related questions and safety of medicines, something similar could be considered for pregnancy and lactation studies as well. I think that's, you know, and I think also if you look at what we have now in methodologies found or worked on in ConcePTION, worked on in LifeSaver, it also provides already a nice toolbox to start generating that data. So I think that really goes hand in hand.
And can you explain in a little bit more detail, maybe for the broader group, what that pediatric incentive is and how that could translate to a pregnancy and lactation incentive?
Yeah, so I think it must have been already kind of 10 years ago, where there was a lot of emphasis on studies in children, because most at the time companies brought their product to the market based on the studies that they did mainly in adults, and there was hardly any data on the use in children at marketing. And both the FDA and also the EMA in Europe have put into place an incentive where companies are forced to do or request or need to do studies in children as well. And they can do it in a staggered approach, so they can first register their product for adults, but still they need to provide a pediatric investigation plan in Europe, for example, where they outline the plan. How are they going to investigate the safety and the effectiveness of their medicine in younger populations? Usually it starts with adolescents, and if there's a need also for younger children, next steps could be to do studies in younger children. And the incentive there is, on the other hand, that gives a prolongation of the patent periods, actually, in the end of the day. And I think something like that is to be considered for pregnancy as well. And I think it's the same vulnerable population that you're talking about. It's children, it's pregnant women, it's the classical populations that are not included in the clinical research. But still, I think, in this time, you cannot just neglect that and say, well, no, we didn't do research in pregnant women. They just need to find out themselves, or they just need to do trial and error. So that's one hand, and also what I do think is that we have a lot of information available. If you look at the electronic health data sets, where we know where is registered if pregnant women are being prescribed medication, that is exactly the data that is already out there, and that needs to be more systematically analyzed as well. And I think that can also be a big push to get more evidence-based information on the safety of those medicines, and just to ensure to ensure, I think, also that it's not an NS1 trial every time a woman makes her own personal decision to take a medicine when she's pregnant. It's a waste of knowledge if we don't take that knowledge further and bring that together with other women that have been facing the same issue.
Yeah. So you're already alluding to two possible action lines, right? As part of Thomas's plan, one is a policy incentive, and the second would be a structure for information provision in a trusted and credible way, right? How does that sound, Thomas? Sorry, Pieter. After that, we'll go to you.
Jasper, I think that is exactly that. Also, third line, which is called which I would call a sustainable resilience monitoring. Because there could be a drug which have been no data at all or not sufficient, and at once somebody find, it's toxic. So that means that you need to reassess it, or is it a drug which has been repurposed or something like that, from both way to positive or negative discrimination, or let's say that, we can call it the prenatal drugs, which may be specifically indicated for patients in pregnancy, for some certain reasons, may be very rare, but anyway, that's kind of happens. So if combining all these records, which already have, which Marjolein has mentioned, with also systematically to go retrospectively and to predict whether the prospective studies needs to be repeated or prospective test needs to be repeated, I think that could be a third line, because that is a part of QMS, it is a part of risk assessment, and I think that's not only concerns the new drug pipelines, but also that which is already on the market, just to see if their safety can be somehow getting more sure that before that.
Yeah, makes a lot of sense. And Miriam, do you want to comment on this topic, or?
Yeah, no, it's yes, no, it's great proposal. So we can try that line of thought for newly introduced drugs, but the key thing is about all those older ones, as Mike was also saying, like, so we should also have a plan for that. And probably there should be like a catch-up plan, no? Like
because there is also the question of exposure. Let's say if the 10 milligrams was, okay, and somebody takes 20, does it present a concern? Okay, if somebody takes 50 milligrams, it's also the question, because it may be that it was considered safe and there's some dosage or administration, and when you change it, or when there is a new indication for that one, and we need to reassess that.
Can we park this for a second and give Pieter the floor, because he's really eager to.
Well, thank you. Yeah, so very interesting discussion. Yeah, I mean, I was going to add something in the same direction, and maybe it's a little bit of an open door, but it's the experience that we got within Work Package 3 is it's really important to get the buy-in from regulatory agencies. In our case, it was EMA, as soon as possible, because you can really learn a lot from them, and also learn how you should communicate about what you achieved. Like within our work package, we were very much convinced about the models that we developed and the potential that was there. But yeah, like you don't tend to see the limitations also, and that's something that they really emphasized on. So, okay, I mean, this all looks very beautiful, but how are these models, or the information that you generate with all these beautiful models, how is that ever going to affect the label? How is that going to impact the safety decisions that you make for women during lactation? And I think that was a very interesting comment. And another aspect I want to mention here is that really hammered on what is the context of use of all these models. You have now tested 10 model compounds at best. In the Eviva model, it was even less. But what does it mean? Can we now test and make predictions for every new small molecule around the corner? Probably not. So they don't want to take. They don't want to give that kind of endorsement. So, and along the same line in terms of context of use, but from a different perspective, in which development stage do you use these models? That's also another aspect that we realized that we need to communicate much better about. Because, of course, in this stage, we also needed to justify very much, and it's still a challenge, that we also need this in vivo animal model. But we probably don't need it until 2040. But at this point in time, we needed to cross-validate the other models and to make progress faster than we would without the model. But that is a very challenging thing, especially to justify and communicate about why you really need this. So this was something I wanted to add to the discussion here.
Yeah, and I think Miriam sure wants to. Oh, sorry, Michael. I think Miriam sure wants to say something about the link to then real-world evidence and public health records when it comes to these outcomes, for example. But Pieter, can you also explain a little bit more? We've talked in the past about a kind of risk assessment or triage system, where that could play a role in that catch-up that we just described, right? So there's many drugs on the market where we currently don't know enough, and we need to start somewhere. So we need to make a kind of preliminary risk calculation.
Yeah,
and you've been thinking about this, right?
Yes, exactly. Yeah, and you need to realize also, and that's I think also the exercise that we went through. You need to realize what is the piece of information that you can generate, and then you need to bring it back to the essence. The models that we developed, for instance, you generate a pharmacokinetic endpoint, you generate an exposure, but that's not going to give you enough information to take a decision. You also need to have the toxicodynamic information merged with that. And then you need to assess also what is the uncertainty about these pieces of information and bring that together. That's another level of challenge that needs to be brought into the equation. But yeah, I mean, I think the good news is it's evolving rapidly, but we need to structurize this better to make it more clear for ourselves, and also to then better communicate about it to be more convincing, and at least have an approach when a new drug is there, or when a new question for another drug is there, that we know where to start. Whereas now, at least a few years back, we didn't have anything, so we didn't even know how to begin, because we didn't have any models at all.
Michael, sorry for interrupting you.
I just was commended, Peter, exactly. That was a point which I was addressed, because what we can, I think, all agree at the moment, we will never know for 100% anything about how the drug is penetrating somewhere and how it works. Mm-hmm. Maybe we can know 99% of that, but eventually that for this big problem, it can be as complex as possible, as Peter just described. But this is one of what's the idea of the LifeSaver approach, to break down it by steps, which could be solved, before talking toxicology. Let's see that how much drugs we actually have in the fetal blood flow versus time and administration, because that gives you an understanding, is it safe or is it not? And if you know for this exposure how it happens, then you can go to toxicology, and then you can see that this dose is sufficient or not, or is the exposure time would be somewhat risky or something like that. So I think that one of this important and difficult problem which Peter just described, we can do it by going step by step and solving that one. And if we have a practical, for example, solution, for example, there is an industry which is like to test some penetration of the drugs through some placental tissues or into the breastfeeding case, and that said, could we, for example, select a panel of certain drugs and compounds, let's say low risk, high risk, whatever, middle risk, which could be used as a comparison tool, because then the any kind of new drugs or old drugs can be tested against them, and then we can rank it to put in some kind of degree of the classification, I don't know, A, B, C, D, whatever, which could be put in a kind of let's say low risk in pregnancy or high risk in pregnancy, something like that. It would be very, very practical outcome.
Miriam, I think you're eager to say something about real world evidence in this context and outcomes.
Yeah, so thank you. Yes, so we have the experimental parts that may predict, and then we should always verify whether that's actually happening by utilizing data. No? So we have concluded that pregnant people do not want to easily go into a clinical trial. Often there have been many incentives to do that and to have them there, but it's not easy. So, but they are using medicines a lot, like 90%, one or more drugs, and we should utilize the data to actually study the outcomes and then relate that back to whatever the predictions were. So I think we should have a comprehensive assessment pipeline or tooling box that we can propose, that then also feeds back maybe into your tools, because we could see actually what is being prescribed at which doses, in which part of the pregnancy is it mostly utilized. Is it first trimester? Is it second? And I don't know whether your tools also would work on second and third trimester, but we can actually see what is the utilization pattern in real life, what are the outcomes of pregnancy in real life, and then try to relate it back in a comprehensive manner.
Yes, and eventually that could be, at the end of the day, be distilled in the OECD technique test guidelines, which could be a good case, which is one of our aim to present a kind of it could be a non-standard method under 211 or 214, whatever test guidelines are used. But they could basically, if that could be accepted as with a certain purpose, knowing its limitations and knowing what it can do and what it cannot do, then I think that would be kind of additional instrument which could be potentially used in all this drug development. We can talk about basically any membrane. We're not only limited in pregnancy. So it could be second trimester, it could be epithelial barrier or whatever. You talk about lungs, or you talk about any kind of things, because eventually, well, you don't need to know how the car works to drive, but you need to know if you turn wheel left, the car turn left, right? So that's the purpose of engineering approach.
Yes, exactly. I lost my thoughts, so I'm going to lower my hand.
So if I would summarize it, right, in this catch-up goal, not Heinz catch-up, but to catch up on all the drugs that are already on the market and identify where additional research is needed, there's effectively two sources. One is the risk modeling based on the in silico approaches, and the other one is the real-world evidence when it comes to possible signals, utilization data, etc. Another topic that I just wanted to address shortly is, and both Michael and Pieter, you've talked about this already extensively, right? Their exposure is one thing, but outcome is another, right? So, Miriam, can you shortly provide a little bit of your view on how you can connect those two things, right? So when we have those exposure via the placenta, or have those identified the exposure via breast milk, how can we then link that in a reliable way to actual outcomes that are representative for those very vulnerable population?
Okay, well, you put me on the about the design. I'm happy to try, but I need help from Marjolein. All right, so I think what we're currently doing with real-world data is that we actually define exposure based on what we find in these real-world data sources, which is a medicine is being prescribed or it's being dispensed by a pharmacy. And we calculate the duration, we estimate whether a woman would be exposed or not in which trimester. So that's the exposure definition, so to say. And then we relate that. We have all type of observational designs because it's not an experimental assignment. So we just observe what is happening. And we typically now use like target trial emulations to then, mimic a clinical trial, but, it's a real world, it's an observational study, but it tries to avoid a lot of confounding and biases. Now, I think if we want to connect what you are doing to this, I mean, we would need to think of how to redefine our exposure assessment. So what I would be curious to see is if we can see what is the dose of exposure and the timing of exposure, could we feed that back in your models and then create an exposure parameter that's based on your models? And I don't know how we need to give it to you, but that we utilize that as an exposure parameter. That's what
we can. Yes, we can. We need concentration as a function of time.
Yeah, but we only have the amount, so we would need to have some
we need
modeling.
You don't have time. You cannot do any kinetics. You need to have time points.
We have time points, and we have the amount, and then we can distill concentration. Using a PK model. Yeah.
We can discuss that, but basically, I think that is theoretically it's possible. I can say.
Mm-hmm. Yeah, and maybe I can add to that. So that's what we have been doing typically also for this project, where you generate the population, and that means that you make some assumptions, but this is also based on data about the underlying physiology. If that physiology is there, then you can actually dose directly into that physiological reality, and then see how this amount distributes over the different volumes that you have defined, which is different organs according to different blood flows. So that is perfectly possible to do that. If that answers your question, Miriam.
Yes, so I think we have a new study design here.
And also one important point, which Thomas was also mentioned about the policy. We talk here about the policy which we are providing, let's say policy advice or development means, but there are also another flow of data which comes from the Commission back to us. For example, in LifeSaver, we have been specifically instructed to do mixtures of the drugs. If you have 2 drugs or 3 drugs, that the system becomes very complex. And you don't know exactly what kind of cross interaction of the DDI comes or whatever. So even from that simplified model, what we have in the just the gut drug transport without yet any toxicological thing, we see that it is a lot of challenges immediately when you go to the more than one drug or more than one chemical. So I don't know from a pharma industrial point of view, how important you see these particular things. Maybe that's not so much, but since we have been asking to do that, what is your opinion about these more complex things? How do you see the challenges there?
Well, I can only confirm. Yeah, sorry, go ahead. Yeah.
Oh, sorry. Yeah, no, I was just, thinking out loud also is that, very honestly, I do think we already have much to gain with, the easy or the simple models already, the low-hanging fruit, because I think there's already a lot of work that should be done is not being done at the moment, and I would think to look at it as a kind of a gradual approach. We start with the basics, and then we kind of should extend actually to more complex systems and see what's next, and also take into account, just coming back to the exposure, is we will first need to learn how the drugs are being used in a post-marketing setting, which combinations of drugs are used, and also not, and we can, as pharma, we can think what will happen, but we will first need to see what is really going to happen in real world before you can, I think, design these more complex models at a certain point in time. But I really do think that there is a need, because, if you look, for example, at chronic illnesses, there's hardly any chronic disease that is treated with only one drug. So really looking into mixtures and combinations, it should be really the next step.
Yes, and I can confirm that the pharma industry is certainly very interested in that. And maybe the good news is that the first application, and that's already accepted by FDA and EMA, of these physiologically based pharmacokinetic models is drug-drug interaction prediction. So essentially what happens then is that they will test the combination of a victim drug together with a drug that is considered a perpetrator, and then a very strong perpetrator. And then you can make predictions, reliable predictions, that has been demonstrated in several studies with other drugs that are less severe perpetrators, and that can even be used as a waiver nowadays for conducting certain drug-drug interaction trials. So I think there is some interesting prior art there in that domain.
And I think that there's another part which comes from the Commission point of view, actually WHO, is that here we're mostly talking about the therapeutic window, but there are also drug abuse. And in WHO has put, especially in developing countries, that the people usually, they don't know or don't care that what kind of specific dose they take. And that is very easy to do overdose, which may have adverse effects. So also that's effects we see that could be potentially studied with in vitro and in silico model in order to see at least some kind of limits which could be, potentially risky or something like that. I didn't dive in that deeply, but basically I can anticipate that this kind of super toxic or supra toxic levels, which is not having therapeutic effect, but which is kind of poisoning concentrations, they also probably needs to be analyzed somehow.
Yeah, very good point.
Yeah, very interesting. Maybe going back to you, Thomas. Sorry, first maybe to the audience. If you have questions to the panel, please raise your hand or put them in the chat. Then I'll give you the floor in a minute. Maybe Thomas, going back to you, right? So you said we need a plan. We need a message. We need an action plan. 1, 2, 3 goals. We've touched upon a few, but maybe you've had some others already in mind.
When we are speaking of objectives that bring to an action plan that we will communicate to somebody, we know to whom we will communicate. From a communicational point of view, in fact, we are talking of problems that are happening from the beginning of pregnancy until the end of lactation. So we are in a period of time which is around 2 years. And we have to make understand that this is a very important period where many things can go wrong and many things can go okay or positively. That could be a communicational approach of what we are trying to integrate. How we call that period, I don't know, the green period, or I don't know. It can have a name, of course.
Yeah. And then if you have you already a view on more the communication plan, right? So you say we need to integrate all stakeholders, right? Then if we want to bring, for example, this idea of this pregnancy investigational plan, or the part where we integrate different methodologies presented here to assess which drugs urgently need to be validated for in the methods that you have developed, right? The drugs already on the market. Do you already have a bit of a view on that communication strategy?
I think what you mentioned, it's okay to integrate. It's actually important to integrate stakeholder, but in regard to communication plan, I think that we have to break it apart, because different stakeholders, they have different ideas. And sometimes you can have to prepare a communication which even member of parliament would understand. So that means something which is very, very kindergarten level. So because they are not specialists at all, but they see that they need to communicate to their voters, so that we take care of your health and everything like that. Because there's also, I think in every country, what we have now, a huge pressure on the burden of the cost, not only a cost of the treatment, but also cost of their supporting measures. So if we have it in the proposal that when we have some adverse effect happening in pregnancy, the cost of that to the society and to parents will go for 15, 20, 30, 40 years ahead, for generations. So basically it has a very big social aspect. And this, I think, one social aspect we often forget it because we are not social scientists. We don't know, we don't think from that point of view, but I think that it's part which we can maybe more actively to involve the social part to get people to understand, because when they go to understand, they will be much more important. Recall, for example, COVID and all these vaccines. Should these vaccines be given to a pregnant women or not? And they could be blown away, all these discussions up to physical fighting about are you for or against. So I think that is what we have to promote: communication to people, communication to the social part, and the communication to the public media. Because that is the amount of useful information what you get from YouTube, it's very, very minor. Regarding to the disinformation or wrong information, you can read it. And I think that is one of the things which we need to communicate to the Commission, because Commission has a very nice video about environmental threat for health from, for example, fluorinated compound or from whatever thing like that. But the videos they have on their website, they has been looked for years, 500 times, 700 times. So the information does not reach the audience. I think this is one of the problems.
Yeah, I think that's a very fair point. Maybe Miriam, you've been involved, right, in COVID vaccine safety. Can you share your experience on the communication aspect on that?
Well, I think, without going into many details because it's not my area of expertise, but what you were seeing is that initially COVID vaccines were indicated for pregnancy in the US, actually. So FDA allowed this, whereas EMA did not allow it. So it was just a different interpretation. And that has led, I think, to a lot of confusion and doubts as well. So how can there be this discrepancy on the one hand? No, and then later on, WHO recommended also that it would be used, and so it's all reverted. But it's based on the lack of evidence, and decision makers take different decisions. So I think that's just a perfect example of what the lack of evidence actually creates. Different decisions and confused citizens.
Yeah, and we see that in this population as well, right? Pregnant women and breastfeeding women. Yeah. Okay. Any other things? I've run through my questions, so not sure if there's anything. Shall we just make a round for any final remarks? Pieter, you want to start?
Yeah, I mean, I can just comment on that. I found this a very interesting discussion, and I hope that we can continue from here to exchange ideas. If you consider the placenta, I'm just talking from my perspective, and you consider the blood-milk barrier, these are just two biological barriers that are different, but in terms of how drugs are crossing, similar mechanisms will apply. How do you deal with that, and how do you exploit that information, how you extract that information to eventually then bring that into an in silico model and then make predictions. So I'm very open to exchange that with the colleagues from LifeSaver in the near future.
Yeah. Thomas, any final remarks from your end?
Yes, well, in general, the perception is that the problems we have been discussing, they are there, outside there, and most of everything is fragmented and dispersed. The good news is that at least we have here two projects which are not fragmented and not dispersed, so probably we would have to build up more the synergies and the commonalities and the complementarities. We are, as I said before, we agree almost on everything, and we are very complementing. So this could be a good kickstart for going further in each of the objectives of both projects and together. So we have to find a way to do that. We need probably to discuss it a bit more.
Cool. Marjolein?
Yeah, well, there's not much I think I can add, because I do think that the commonalities between the two projects really kind of indicate or hopefully also give this as a starting point to combine forces, and not only on the operational parts, on the development of methodologies, but even more important, I think, on driving the agenda for policymakers in Europe to keep this topic high on the agendas. And because our project is ending in 7 months from now, I really feel that it would be a waste of all the work and all the effort and all the dedication of the people that have been working on that to just go apart and say thank you, but to actually use this as an opportunity to give it a more sustainable way forward as well. And I think with combined forces, you're always stronger than on your own. So I'm really looking forward to further explore how we can collaborate and intensify the working together.
Yeah. Would you like to share a couple of words on how you see this beyond the project, right? To make sure that all those energy and interactions are not lost? Marjolein?
Sorry. I saw what you said, Miriam. I was confused. And I think that from a conception perspective, that is really like the number one on our to-do list. And how are we going to move forward with that? I do think that they're different, and that's actually just reflecting the discussions that we had today. There are different streams that need to be worked on in parallel. So it's both policy, raising awareness, keeping it on the agenda. It's also making sure that we have the methodologies, that we have the methods to generate this evidence on the medicines, because that goes hand in hand. If you can provide a solution to a problem, I think your case is much stronger if you're only identifying a problem. And also I think what I just said, and also was reflected in the discussion actually about the COVID vaccines just now, is how do you get that information in a uniform and evidence-based way to healthcare professionals and patients. So I think that's, for me, the three angles of this topic that really need to come together and need our continuous, I think, effort and work on that.
Very nice summary. Thanks. Michael?
Okay, I can only agree with what has been said. I think that is very, very good. And then in the case that if we organize it in any kind of a new project or idea, we need indeed to involve different level of stakeholders. We need to have social people. We need to have media. We have to have this and this. And regarding the evidence, what Marjolein was mentioned, I think that may be one of the possible way. Actually, we use it already for many years. Is a kind of shuttle between, making tests and getting toxicology, analyzing, going back, iterating that before we reach the level of evidence which we can present, and then said, okay, this is something like that. So in LifeSaver concept, we tried to simplify the things as much as we could, because if something is very complex, I don't think that the industry would be very happy to take it if you take a lot of efforts and everything. So that has to be practical solution, and it has to be, whether we talk about digital part involving, for example, digital health records. I had experience in orthopedics. So basically when we heard, I just tell you some anecdote on that. So in orthopedic conference, we heard about from the National Joint Register in the UK. It was a very nice presentation with a lot of statistics saying that, okay, if you are over 65 and you fell down and you break your hip, then your restoration will be, like this and this and this. And after that, there was a purple from the register saying, oh, after our investigation, we find out that in this joint register, 55% of all the data are wrong. The leg was marked wrong, the gender was marked wrong, the name was wrong, like something like that. So how do you actually know that the data what you have that you can trust, especially if the data has been made 15 years ago or 10 years ago, which you cannot even find what was the real thing. So I think that whether we have the data for whatever sources, that would be extremely good thing that we can provide an extra validation that this data either repeated or somehow validated to know to which extent we can trust them, because then we have much more stronger starting point for collecting evidence.
I think that makes a lot of sense. Anything you would like to add, Miriam, or any final remarks?
Well, I think a lot has been said, and I would concur that we need to continue talking and making plans actually, and maybe more detailed in how this can fit together. I don't know whether that can be a new project or whether we can continue in another way, but let's look at that. So there's an invitation, I think, also from our end to continue this engagement, and I'm very excited and thank you for the opportunity and reaching out and finding this synergy. So I think, at least from my end, and I also hear Marjolein from the ConcePTION, and we would be very much willing and keen to go forward, and also of course Pieter, like the different streams of So thank you.
Thank you.